Good morning, all of you. In the last class, we has been discussed regarding merits of vertical classification, demerits of linear classification. We has been discussed, and today I am going to start with the first kingdom is Munira. Here, the characters of Munira. What is the main characters of Munira? Silent features of Munira. Here, bacteria is a solo organisms in Munira. These all are only prokaryotic organisms are grouped or belong to the Munira kingdom. Next, the main character is bacteria cell wall. Bacteria cell wall. As well as you know that in the previous class also we has been discussed. The cell wall is made up of peptidoglycan or murin. Peptidoglycan or murin. Next, the next main character is, and we know that the nucleus is absent. It is a primary nucleus or nucleus is completely absent. So that's why chromosome, only one chromosome, single chromosome is present, which is present in cytoplasm, So that's why it is also called as the DNA is naked DNA or open DNA. And one more main feature of Munira is double membrane cell organelles are completely absent. And respiratory enzymes present in plasma membrane. And here, generally in eukaryotic organisms, cytoplasm movement we can observe. The movement of cytoplasm is called as cyclosis. But here, cyclosis is absent. What is meant by cyclosis? Simple. The movement of cytoplasm, maybe clockwise movement or anti-clockwise movement is called as cyclosis. But in bacteria, cyclosis is absent. Next, here in bacteria, we can notice ribosomes. So these ribosomes, we know that mainly two types of ribosomes we can notice. One is 80S ribosomes, second one is 70S ribosome. In bacteria, mainly 70S ribosomes we can notice. S means already you know the Sedberg unit. It is a scientist name. S means Sedberg. Next. And here in bacteria, one more main feature is additional chromosomes are present. Such additional chromosome is called as plasmids. That is called as plasmids. Here one chromosome is present, one single chromosome is present. Clear? Beside the single chromosome, other than chromosome material or additional chromosomes are present. Such additional chromosomes is called as plasmid. It is also one of the main features of bacteria. Next, and reproduction. Reproduction, you know the target. Reproduction is mainly sexual reproduction, asexual reproduction. So here in bacteria, actually true sexual reproduction is absent. So that's why it is also called as parasexual reproduction type. It is not a true sexual reproduction. Next, asexual reproduction. Asexual reproduction binary fission. Asexual production with help of binary fission. Next. So this is actually the main characteristic features of kingdom Munira. And here one more character is here A mitosis. Through A mitosis, the cells are going to get developed. Then, what is the major difference between mitosis to amitosis? In mitosis, spindle fibrae is formed. So this spindle fibrae is going to be useful to separate the chromosomes. Equal distribution is occurred if mitosis is involved. In this case, in amitosis, here spindle fibrae is not going to form. So that's why here chromosomes are equally not distributed. Clear? So this is amitosis, also one of the main characters of Munira. Next. Here. Next. Now we are going to discuss regarding bacteria. Nothing but eubacteria. In the last class we have been discussed the difference between Archaebacteria to Eubacteria. Now I am going to start with Eubacteria. You know that U means what? U means true bacteria. Here, true bacteria. The cell wall is made of peptidoglycan as well as murine. Next. So these bacteria are first discovered by Anthony van Leeuwenhoek. These bacteria are first discovered by Anthony van Leeuwenhoek in rainwater as well as like pond. He is discovered or he is observed the microscope. He is discovered the microorganisms with the help of microscope. The help of microscope. And these organisms he was called as animalcules. It is called as animalcules. And he wrote a book and he is published in 1695. He published one book. The book name is Secrets of Nature. Secret of Nature. In this Secret of Nature, he is discussed regarding animalcules. Next. Later, Linnaeus is observed the same microorganisms. He is called as vermis. V-E-R-M-I-S. He is called as vermis. Later, Louis Pasteur. Later, Louis Pasteur is observed. So these micro living organisms. Actually, micro living organism, the term is first used by Pasteur. Micro living organisms or microbes. So such word is first used by Louis Pasteur. And the Louis Pasteur is also considered as father of microbiology. He is also considered as microbiology. Micro means small, biology means you know the living organisms. Next, Louis Pasteur he was first introduced aerobic bacteria as well as anaerobic bacteria. And you know that aerobic bacteria that will survive only in the presence of oxygen only they can survive. Anaerobic bacteria that can survive in the absence of oxygen also they can survive. Less amount of oxygen also sufficient to survive of this bacteria. Anaerobic bacteria. Next, Louis Pasteur, he is also proposed a germ theory. So according to the germ theory, here the disease, almost a maximum disease are caused by only microorganisms. Diseases are caused by microorganisms. Next, after that, one of the famous microbiologist is Robert Koch. Robert Koch was proposed the postulation theory. Is proposed the postulation theory. Postulation theory means what here? Is give the criteria to determination of microbes. To determination of bacteria. Determination of bacteria. So the criteria is the determination here is given for terms are given according to the Robert Koch. He says that we can generally the bacteria are present in a diseased individuals and bacteria are absent in a healthy individuals. The first point is the bacteria are not present in healthy individuals and bacteria are present in only diseased individuals. Next, second point of postulation theory. So these bacteria are we can isolate. The second point is we can isolate the bacteria from diseased individuals and also we can culture the bacteria by using of plant medium. So his first scientist, he was artificially considered the culture the microorganisms. Further, he got the Nobel Prize. Next point is here. These bacteria, what we culture these bacteria? These cultured bacteria, if we inoculated or we injected to the animals or humans, so such individuals also be suffering from diseases. Third and last one, we can re-isolate the bacteria from that diseased person and such are exactly identical to the original bacteria. So this is actually postulation, the criteria of determination of bacteria. So this is proposed by the postulation theory proposed by the Robert Koch. What is the first point? Bacteria are they are present. We can notice only in diseased individual. These bacteria are absent in healthy individuals. Next point. These bacteria we can separate from the diseased individuals and also we can culture by using of plant medium. After culture, if these bacteria is going to introduce or injected or inoculated to one individual, such individual should be suffer from the disease. And fourth, from the diseased person, we can re-isolate the bacteria and that is exactly equal to original bacteria. So this is what is telling the postulation theory given by Robert Koch. Now, only one scientist, his name is Ehrenberg. Ehrenberg, he was first using the term bacteria term, it is first used by Ehrenberg. The bacteria term, it is first used by Ehrenberg. Okay. So this is some of history regarding bacteria. Next, distribution of bacteria. Distribution of bacteria. So these are the most abundant organisms. Most abundant microorganisms on the earth. Occurrence is the mostly occurred everywhere. The mostly occur everywhere. See, the bacteria occur the present in soil as well as the present in water also, and air also is present. In soil also you can notice number of different types of microorganisms we can notice. The first one is, for example, Nitrosomonas bacteria, as well as Rhizobium bacteria. So these two bacteria occurred in soil. Next, water. In water also we can notice bacteria. E. coli, Escherichia coli, it is occurred in water. In air, Staphylococcus is a bacteria, it is occurred in air. And some of the bacteria have the capacity to survive in a high temperature also. So such bacteria we can call as thermophilic bacteria. They can tolerate nearly 75 degrees Celsius temperature. They can nearly tolerate. These are Thermus example. Thermus aquaticus. It is thermophilic bacteria. It can tolerate high temperature also. Next, and some of the bacteria they can tolerate low temperature also. Such are called as psychophilic bacteria. They can survive in less than 0 degrees Celsius also. They can survive. Psychrobacteria. It is an example. Next, and some of the bacteria they can survive within the human being also. So such as intrinsic bacteria. So for some bacteria they can survive within the stomach also, in small intestine. It is also E. coli bacteria, Escherichia coli. And some bacteria they are in symbiotic nature. What is symbiotic nature? In this symbiotic nature, here it is a mutual relationship between two organisms. Symbiotic relation means what? It is a mutual relationship between two organisms. In this mutual relationship, both have the benefit. So such a relationship is called as what? Symbiotic relationship. It is called as symbiotic relationship. Here, Rhizobium bacteria. Rhizobium bacteria, that is associated or symbiotic with legume plants, like bean plants. Beans are belong to leguminosa or leguminous family. So this rhizobium bacteria is going to associate with legume roots. So here, so this is actually the distribution as well as occurrence of bacteria. Next, size of the bacteria. Bacteria size. The average size of bacteria is 0.5 million to 5.0 mm. Actually, in some book it is given 2.0 mm, 3.0 mm is given. But the recent information and exact information is 0.5 mm to 50 mm. Next, in bacteria, smallest bacteria in Monera, smallest bacteria in Monera kingdom. It is Dialistis. Dialistis, they say smallest bacteria in Monera kingdom. The average size is 0.15 medium to 0.30 mm. Next, one more bacteria is there. The name is mycoplasma. Mycoplasm, it is smaller than dialistic bacteria. The average size of mycoplasm is 0.1 medium to 0.3 mm. Here, mycoplasma is a smallest microorganism in a living world. Next, the largest bacteria is largest bacteria is Theomargarita namibiensis. Theomargarita genus, namibiensis is a species. Here, the average size of Theomargarita namibiensis is 100 million to 300 million. So this is the largest bacteria in Monera. Next, and one more bacteria is the largest filamentous bacteria. The largest filamentous bacteria is Brigiola. Largest filamentous bacteria is Brigiola. So this is various size of the bacteria. Clear? The smallest bacteria, actually Mycoplasma, it is also considered as bacteria. It is also considered as a bacteria. So this is the smallest size of bacteria. Largest bacteria is Theomargarita namibiensis. Clear? Next, different shape of bacteria. Different shape of the bacteria. The basic shape of the bacteria are mainly four types. The basic shape of one is spherical shape, and second one is rod shape, third one is comma shape, and fourth one is spiral shape. Spiral shape. And one more type is there. Fifth one is pleomorphic. Pleomorphic bacteria. What is meant by pleomorphic bacteria? Pleomorphic bacteria actually it have the capacity to change their shape, not a particular shape. Example is Rhizobium. Example is a Rhizobium. Clear? Next. First one is spherical bacteria. Spherical bacteria. Normally it is round in shape. It is a round in shape. And it is do not contain any flagella. Flagella is absent. So that's why it is also called as Kochi bacteria. Non-flagellate. It is non-flagellate. Okay. So this spherical bacteria, nothing but Kochi bacteria, it is again divided into further types. Is Monococcus, Diplococcus, Streptococcus, Staphylococcus, and the last one is Sarcinae coccus. These are the further types of spherical shape. Simple. Monococcus. Mono means what? Single. Here only one single cell is present. Only one single cell. Example is Micrococcus bacteria. Micrococcus bacteria is example of monococcus. Next one is Diplococcus. Diplo means two bacteria. Example is Diplococcus pneumonia. Diplococcus pneumonia is an example for Diplococcus. Next, Streptococcus. Here, these are looked at as chain like diagram. Example is Streptococcus. Streptococcus. Next, Staphylococcus. Here, large number of bacteria that are arranged in irregular arrangement of bacteria. So such a shape is called as Staphylococcus. Example is Staphylococcus bacteria. Staphylococcus bacteria. Last one is Sarcinae type. In this Sarcinae type, the bacteria are nearly 8 bacteria are arranged to form like as cube-like structure is formed. Cube-like structure is formed. So here the example is Sarcinobacter or Sarcinobacter. The diagram of bacteria is like 3-dimensional view. We can notice 4, here one is 5 and 6, here 7 and here one more is 8. Like arrangement is occurred. Here this is a types of different shapes of the bacteria. So to come to the question here, So here the bacteria here is single, and in this case you can see number of bacteria are there. So why these bacteria are grouped? Simple. The answer is here. In Diplococcus, Staphylococcus, and Streptococcus, Staphylococcus, and Staphylococcus. Simple. When after mitotic division, two cells are going to form. These two cells are going to separate. But in this case, what happened? After the mitotic division also, the daughter cells do not get separated. So that's why different shape of the bacteria are formed. Clear? Have you got the point? Generally, after mitotic division, two daughter cells are formed. But in this case, after mitotic division, the daughter cells do not get separated. So that's why. So this different shape of bacteria are formed. Clear? Streptococcus, Staphylococcus, sessile, giant, irregular, cuboid shape or cube-like structure. So this is a different types of bacteria. This will be coming in a spherical shape. Next one is what? A rod shaped. A rod shape. So here this is a rod shaped bacteria. Example is Bacillus. Example is Bacillus. Next one is comma bacteria. First major problem is comma shape. Comma shape. Here, like this is a comma shape bacteria. Example is Vibrio. Vibrio bacteria. Clear? And last one is spiral shaped. Spiral shape. Here, these bacteria are off-coil bacteria. Like so these are spiral shaped bacteria. Spiral shaped bacteria are example is Spirillum. Spirillum is a spiral shaped bacteria. Next, flagella. So flagella or flagellum, which is useful for time. It is useful for movement of the bacteria. Movement of bacteria. Flagella, which is arise from basal granules of plasma membrane. Plasma membrane. Generally, flagella is made up of flagellin protein. Flagella is made up of flagellin protein. And here, based on flagella number, bacteria we can divide into following categories or following types. The first one is A. Trichus, Amphitrichus, Monotrichus, Amphitrichus, Lephotrichus, and fifth one is Cephalotrichus. The last one is Peritrichus. Last one is Peritrichus. First one is Atrichus. Atrichus means flagella is absent. Flagella is absent. So such bacteria we can call as Atrichus bacteria. This is single. We can also call as Kochi bacteria, Atrichus bacteria. Example is Lactobacillus. Lactobacillus. Next, Monotrichus. Here, any one of the side, only one flagella is present. Only one flagella. Only one side. Only one side. So that is called as monotrichous. Like for example, this is one bacteria. From this side, it is containing one flagella. Clear? So such is called as what? Monotrichous. Example is Vibrio. Example is Vibrio. Next one. Amphitrichus. Amphi means two. The two flagella are present either side. In both sides, they are containing one flagella. So such is called as Amphitrichus. Two flagella are present. Two flagella are present from both sides. From both sides. So such type is called as Amphitrichus bacteria. Here, simple saying this is a bacteria. Here, from both sides are containing two bacteria. So such type of bacteria we can call as what? Amphitrichus bacteria. Next, example is Nitrosomonas. Nitrosomonas bacteria. It is the best example for Amphitrichus bacteria. And this one is Cephalotrichus. So in this case, what happened here? Group of flagella is present any one of the side. Group. It means maybe more than two flagellas, two or more flagellas present from one side. Here, one side. So such type of flagella is called as Cephalotrichus bacteria. So here the simple diagram of Cephalotrichus is here. This is a bacteria. Here from this side, from only one side, they are containing more than 2 or 3, 4 like flagella are present. Example is Pseudomonas. Pseudomonas bacteria is an example for Cephalotrichus bacteria. Next, Lephotrichus bacteria. In this case here, same like 2 or more flagellas are present, but both sides are containing group or 2 or more flagellas are present. So such type of bacteria we can call as, based on flagella, it is called as Lephotrichus bacteria. Example is Spirillum. Example is Spirillum. The last one is Peritrichus. So in this case, Peritrichus. In this case, what happened? Here, number of flagella are present, complete surrounding the bacteria. Here, number of flagella are present surrounding on bacteria. Like this is a bacteria. Complete the bacteria surface is containing flagella. This type is called as Peritrichus type. Example is E. coli, Escherichia coli, E. coli, genus and species. So this is actually sixth type, Peritrichus type of bacteria. So this is actually today's class. Clear? The next tomorrow we are going to start with the structure of bacteria we are going to discuss in the next class. Thank you.